Hi everyone. In this experiment, you're going to learn the effect of various salt concentrations or osmotic pressure on the growth of microbes. So in this lab, we're basically going to take bacteria and grow it on agar plates with various salt concentrations, and you're going to incubate them. Then we're going to see whether your bacteria is able to grow on high salt concentrations. So for this experiment, the first thing you need to do, like always, is wash your hands, clean the surface you're going to work on. The things you're going to need is in your lab kit, there's a Ziploc bag, and the Ziploc bag says osmotic pressure. It has 4 nutrient agar plates in it, so take out those 4 nutrient agar plates. You're going to need your subculture of bacteria that you all made. So this is your bacteria growing on an agar slant. And then I want everyone to have 4 inoculating loops. So make sure you have 4 of these because we're going to streak 4 plates. And then finally a Sharpie to label your plates with. So when you look at your plates, they're going to be labeled, and one of the plates will say Na, or what looks like Na. This is just nutrient agar. One of them will say 1%. This is nutrient agar with 1% sodium chloride in it. One of the plates will say 15%, and one plate will say 10%. So what I want you to do is I want you to lay them out in front of you. Lay them out in the order of Na, then 1%, then 10%, then 15%. So we're starting from basically no salt to 15% salt. And then what I'm going to do is what I always do is label your plates. So I'm going to put today's date on my plate and always label the sides. And then that's basically actually all I'm doing. I'm just putting today's date on all of them. You can put your name on it, but you're home, so they're your plates. Okay, and the way we're going to do this experiment is I grew bacteria here, so your subculture has bacteria growing. I want to take this bacteria and I want to grow it on these 4 plates with various sodium chloride concentrations. Then I want to incubate it and I want to come back and see this bacteria, is it able to grow on high salt concentrations? Because I already know it can grow on nutrient agar. I just want to see, will it grow on high salt concentrations? So the way we're going to do this experiment is I want you to use a new inoculating loop for every single plate and start with the plate that has no salt to the highest salt concentration. So this is what I'm going to do. I'm going to hold your slant so that it's like that, it's slanted, hold it like that, and then open it. And I've already opened my inoculating loop at the stick side. Remember, don't touch the loop because this is where we want to pick up the bacteria. Now, this is where students sometimes get confused. So I see a lot of bacteria on my slant. You need the tiniest amount possible. Don't go like that and scrape off a lot of bacteria. If you can see bacteria on your loop, then you took too much. If you did make that mistake, it's okay, but just for the future, you need very little. You don't need to go like that and take a lot. So I'm going to go inside my slant and just pick up a little bit of bacteria, which I did here. And then I'm going to go to my Na plate and basically just go like that. Just streak it like that. That's all I'm asking you to do. There's nothing special here. We're not doing streak plate technique. We're just going like that because you already have a pure culture. So that's my Na plate. And then now I'm going to do my next plate, which is the 1% sodium chloride. So I'm going to open a new loop. And then once you have your new loop, take a little bit more bacteria. So there, I grabbed some bacteria. I can't see it. You really shouldn't be able to see it. So again, if you picked up a lot and you can see it on your loop, you have too much. Then I'm going to do my 1% plate. So just putting it all over my nutrient agar plate that also has 1%. And then I'm going to do the same thing with my 10% plate. So take some bacteria and streak it on my 10% sodium chloride plate. And then finally my 15% plate. So again, a new inoculating loop. And then again, take some bacteria and streak it on the 15% sodium chloride plate. Okay, now once you've done that, you can close your subculture. Please keep this. We're gonna do it. We're gonna use this bacteria for future experiments. So you can just set that aside. So just keep it at room temperature somewhere high where no one can reach it. And then I have my plates. Remember, we always incubate agar side up when we're talking about plates. So I have my Na plate, just nutrient agar, my 1% sodium chloride plus nutrient agar, my 10% plate, and then my 15% plate. I put them all, make sure they're all agar side up. So have them like that. And then what I'm going to do is I'm just going to incubate them at room temperature, keep them like that probably for a couple of days. And so you've done the experiment. Now I'm just going to talk about the theory behind this. So everyone in here learned about hypertonic conditions. So hypertonic solution, such as very salty water or very salty agar, what it does is when you put cells in hypertonic conditions, the cells lose water because water always goes where there is more salt, and the cells shrivel. So for most cells, for most bacteria, when you put them in hypertonic conditions, they die. So salt is a good way to control microbial growth. Now there are some bacteria that fall into two categories. There's facultative halophiles and obligate halophiles. A halophile just means an organism that loves salt. So halo means salt and phile means loving. So a facultative halophile is an organism that can tolerate various salt concentrations. So for example, if I come back the next day and I see that all of my plates have bacteria growing on them, I would classify that bacteria as a facultative halophile, meaning that that bacteria likes salt, it can tolerate it. If I come back and the only plate that has growth on it is the plate that is the highest salt concentration, I would classify that organism as an obligate halophile, means that that bacteria, for it to grow, it's obligated to have salt. And then finally, for most bacteria, I envision that you're only going to see growth on the Na plate or the plate with very little 1% sodium chloride, and you won't see growth at the other plates, and that's because they're hypertonic conditions. Most bacterial cells shrivel in hypertonic conditions because they lose water to the solution. So just keep that in mind, and hopefully you'll see some interesting results.